In nuclear transport, we will talk about proteins entering the nucleus and proteins leaving the nucleus. We will talk about import as well as export of proteins. Okay, so nuclear envelope is a double membrane structure in which outer membrane is continuous with endoplasmic reticulum, and when ribosomes are attached with it, we call it a rough endoplasmic reticulum. You can see here are numerous nuclear pores which are made up of nuclear pore complex NPC. A nuclear pore complex is a large protein complex which is made up of 30 different nucleoporins. It is cylindrical structure as you can see here, and it is having a octagonal radial symmetry. It controls the bidirectional transport between nucleus and cytoplasm or cytosol, and this bidirectional transport is usually of fully folded proteins. As and this nuclear pore complex has three subcomplexes you can see in this figure. The first is cytoplasmic filaments. You can see these are the cytoplasmic filaments protruding towards the cytoplasm. There is central transport channel, and there is nuclear basket which is protruding towards the nucleus. For cytoplasmic filaments, these filaments are extending into the cytoplasm, and they also contain nucleoporins for cargo recognition. The main feature of this central transport channel is these what we call FG repeats. F stands for phenylalanine and G for glycine. This is FG motif, and these are the specific sequences which will interact with the transport proteins when they will translocate through this channel. Okay, we can say that this FG repeats can make a selectivity or a permeability barrier. And finally we have nuclear basket which allows the final translocation of the proteins. So this is a much better diagram of nuclear pore complex. As you can see, we have cytosolic fibrils which are protruding towards cytosol. We have nuclear basket protruding towards nucleus. We have membrane ring proteins attaching this nuclear pore complex to plasma membrane. We have central channel in which we have these FG repeats, phenylalanine and glycine, and you can see this is protein bounded with cargo which is entering and interacting with these FG sequences. That's how the transport works. And we have some scaffold nucleoproteins as well. So this was a nuclear pore complex or NPC. Now we will talk about proteins transported to nucleus. Mainly the proteins which are transported to nucleus are synthesized by the ATS ribosomes, which are membrane free ribosomes, which will form proteins. Proteins get folded, and in fully folded conformation these proteins will be transported to nucleus through gated transport. Now depending upon the size, that mode of transport will differ. If there are macromolecules of size around 40 kilodaltons, they will be transported through simple diffusion, we call passive transport. They, examples are these ions, they may be nucleotides, maybe small metabolites. There is no selectivity barrier for these molecules, and they can be easily transported through passive transport in energy independent manner. If the size of the molecules is greater than 40 kilodaltons, we will transfer these through active transport, which means that there will be a utilization of energy. Now for the active transport, proteins must have a specific signal sequence we call nuclear localization signal sequence. If the protein is entering nucleus, we have NLS for import, and if the protein has to leave the nucleus, we have NES, which is nucleus export sequence. These specific sequences are being recognized by signal specific receptors, and these specific receptors are proteins of family karyoferins. There are two main proteins for two main types of transport. First is importin and second is exportin. Importin is used for import and exportin is used for nuclear export. Now, after the recognition with a receptor, there will be translocation through a translocation channel. We already talked about this translocation channel, which was NPC, Nucleopore Complex. And nucleopore complex is made up of different types of nucleoporins. And finally, there will be regulation. The energy is provided by this RAN GTPase cycle. We will see all these processes in details in a little while. Now, to summarize the process, we have characteristics, and we are comparing the nuclear import with nuclear export. The size must be between 40 to 60 kilodalton for nuclear import, and same with the nuclear export. If the size is between 40 to 60 kilodalton, we are talking about passive transport, and if the size is greater than 60 kilodalton, we are talking about active transport. For import, we have NLS, which is lysine and arginine rich sequence. We talked about that in previous video, introductory video. If the signal is for nuclear export, it will be leucine rich hydrophobic sequence. The proteins will be transported in the folded state. Receptors will be importins for import and exportins for export. The energy will be provided by RAN GTP gradient. And examples, you can take, for example, histones into the nucleus. We have various transcription factors. We have RNA polymerases for the transcription, and for nuclear export we have rRNA binding proteins. We have some tRNA export factors. Now let's talk about nuclear localization signal. Nuclear localization signal is composed of basic amino acids, which are positively charged amino acids, mainly lysine and arginine. Why basic amino acids? Because the basicity of these amino acids will allow their binding with import specific receptors for this nuclear localization signal. It can be anywhere in the protein sequence, but mostly it is internal and it is non-cleavable. In some rare cases it can be N-terminal or C-terminal, but it always, in almost all the cases, it remains exposed on the protein surface so that importins can recognize and bind with this sequence, and the process of translocation will continue. Talking about sequence types, it can be monopartite or it can be bipartite sequence. What is the difference between these two? Look at this T antigen. T antigen is monopartite sequence, means it is the repeat of lysine and arginine. You can see right here, lysine, 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 arginine and lysine. This is only a one patch or a one single strand of this sequence. This is monopartite sequence. This is a single 7 to 8 amino acid sequence, and most common example is T antigen in SB40. But in case of a bipartite sequence, we have, for example, this lysine arginine repeat and we have lysine repeat. We have two distinct sequences separated apart by a spacer. This is what we call a bipartite nuclear localization signal. Examples are nucleoplasmins, and the sequence is as shown. Recognition of nucleus or nuclear localization signal is carried out by importin alpha. Importin alpha can bind directly to this signal sequence, or importin beta. Importin beta cannot directly bind to this sequence, but importin beta interact with importin alpha. So importin alpha can also act as an adapter sequence. For energy and directionality, as we talked earlier, it will be driven by RAN GTPase gradient. Inside the nucleus, there is a higher concentration of RAN GTP, and this higher concentration of RAN GTP triggers cargo release. As in cytoplasm, there will be lower concentration of RAN GTP, and importins will free themselves to bind to new cargo. So this is the comparison table of monopartite and bipartite nuclear localization signals. We already talked about this in previous slide. Now we will talk about nuclear export signal. Typically, the nuclear export signal consists of leucine rich hydrophobic sequences, and it is around 8 to 15 amino acids long. The consensus motif is this symbol here phi. This phi represents the hydrophobic residue. It may be leucine, it may be isoleucine, it may be valine, phenylalanine or methionine, but most commonly it is leucine. X can be any amino acid, and these hydrophobic residues form a hydrophobic phase that fits into CRM1 binding group of the exportin protein or exportin transporter protein. But is the location? The location is mostly internal and non-cleavable, though in some rare cases it can be on either terminus, but mostly it is internal and it is non-cleavable. It is recognized by exportin protein, or exportin, we can say exportin is the receptor to nuclear export signal, and for specific exportin there is CRM1, which is exportin 1 we talked about earlier right here. It is the primary nuclear export signal receptor. What is the energy source? Energy source is same as usual. RAN GTP hydrolysis powers this directionality, and RAN GEF, which is guanine nucleotide exchange factor, ensures that there will be high concentration of RAN GTP inside the nucleus, and RAN gap will be in the cytoplasm. It ensures the hydrolysis of RAN GTP, and it will convert this RAN GTP into RAN GDP. Okay, this is the summary of the previous slide where we talked about the nature of the sequence, which is leucine rich hydrophobic 8 to 15 amino acid long sequence. We talked about specific motifs. We talked about this phi is a specific sequence which can be either of these sequences. Most commonly it is leucine, and X can be any amino acids. It is mostly internal and non-cleavable. Proteins are exported in folded state. About receptor CRM1, and energy source is RAN GTP hydrolysis. Examples are P53 protein. We will study this in more detail in cancer biology, and this PKI. Now, this table represents a localization signal for import versus nuclear export signal for export. This is a comprehensive comparison between these two signals. You can go on your own. Nuclear transport receptor proteins. These are the soluble proteins mediating import and export of macromolecules. In this section, we will basically talk about the importins and exportins for import and export respectively. It belongs to, or they belong to, karyofurin family, and they can transport proteins in folded state. They use RAN GTP cycle for their energy source and directionality. What are the key members of these nuclear transport receptor proteins? I talked about two key members. One is importins and second one is exportins. In importins, there are several types of importins. For our interest, we will study about importin alpha and importin beta. As I already stated that importin alpha can directly bind, or it can act as adapter for importin beta. In case of exportins, we talked about CRM1, which is exportin 1 receptor, which is the major exportin receptor, and we will also talk about exportin T, which exports mature tRNAs, exportin 5s, which exports pre-miRNA hairpin loops, AS, which exports importin alpha after cargo release. It is for recycling of this importin alpha. Now you can go on your own with this table, which represents almost the same information here. The new information is like importin 7, which transfers histone, H1 or some ribosomal proteins. We have syndarptin, which transfers snRNPs, which is U1, U2, U4, U5. These are, if you remember, these are proteins which are used in the splicing of mRNA. Export, we already studied CRM1, exportin T and exportin 5 already in this slide. So you can go on your own with this table as well. In case of importins, we can say that they are involved in active import of proteins, because the size of proteins is more than 40 or 50 kilodaltons. There will be utilization of energy by RAN GTP gradients. So importins are for active import of these proteins. They bind to NLS on cargo protein, When they pass through this translocation channel, they interact with Fg receptors on these nucleoporins. What we can take away from this slide is that importin alpha recognizes the nuclear localization signal and importin beta is one which interacts with this Fg nucleoprotein Fg motifs. And this importin beta recognizes importin alpha through a specific IBB domain. For regulation, we will talk about three types of regulation. First is masking or unmasking of NLS. If the NLS is not visible on the surface of protein, there is no way that importin can bind to that protein and that protein cannot be transported. In case of there can be post-translation modifications, like there can be phosphorylation or simulation, alternative importins, different importins or some isoforms of importin alpha for specific cargoes. That's how the regulation of these importins takes place. Here are some examples of proteins which are transported with the help of these importin receptors. Again, here is comparative table of importin alpha and importin beta. We already talked about all of the previous slides. Now let's talk about exportins. Exportins mediate active transport of proteins and RNAs out of the nucleus, just like importins does. They recognize nuclear export signal and they are also RAN-GTP dependent. They are the members of importin beta superfamily and they operate only when the RAN-GTP levels are high enough inside the nucleus so that they can ensure the directionality. Again, regulation is very similar but we talked about earlier. The signal masking, unmasking, post-translation modifications. There are some viral manipulations in case of viruses like HIV, which exports the CRM1 receptor to export the viral RNAs, and there can be pharmacological inhibition with some drugs. The main drug you can remember is leptomycin B, which covalently binds to this receptor and blocks the export. Then we have some exportins, their function and some comments. You can go on their own. This is the CRM1 exportin 1 we talked about in the details in previous slides. Exportin T, T represents tRNA, it transfers tRNA, it is highly specific. In case of Cas, this is cellular apoptosis susceptibility protein. It exports importin alpha back to the cytoplasm. It is helping in recycling of the importin alpha receptor, and in case of exportin 5 it is exporting pre-miRNAs. In case of exportin 6 it is exporting actin. In case of exportin 7 it is less specific and exports a wide range of proteins. You can go on your own for this slide as well. Now we are coming to general mechanism of transport. We studied this mechanism in previous lecture where we stated that there has to be some sort of transporting signal on the protein. In case of nuclear transport, this signal is either in the form of nuclear localization signal or nuclear export signal. This signal is recognized by the receptor. In case of nuclear transport, we have importin or exportin as receptors. Now after this recognition, this importin or exportin will go through nucleoporin channel where they interact with Fg motifs and energy source is RAN-GTP. Now we will talk about RAN-GTP and RAN-GDP compartmentalization. In cytosol there is higher concentration of RAN-GDP, and why is this there higher concentration of RAN-GDP? Because there is higher concentration of RAN-GAP. There is high concentration of RAN-GAP. RAN-GTP cannot exist freely, it will be hydrolyzed into RAN-GTP. Whereas in case of nucleus there is higher concentration of RAN-GTP because there is higher concentration of RAN-GEF, guanine nucleotide exchange factor. Now this nucleotide exchange factors will convert this hydrolyzed GDP into GTP. This is diagram from textbook itself. As you can see inside the nucleus, the form which is existing is RAN-GTP, and This GTP is provided by RAN-GEF. The most common RAN-GEF is RCC1. RCC1 is providing this GTP to this RAN-GDP. GDP will dissociate and GTP will bind to this protein, and there will be major concentration of RAN-GTP. This RAN-GTP will pass through uh, its specific receptors and come to cytoplasm. In cytoplasm, there is higher concentration of RAN gap, and this RAN gap will dissociate or hydrolyze the phosphate, and there will be RAN GDP concentration. Now this RAN GDP will cycle back to the nucleus. In nucleus we have RAN GEF, which again transports the GDP, which removing the GDP, and that's how this cycle operates. Now we will talk about the detailed mechanism of nuclear import. See here, there is cargo. Cargo has nuclear localization signal. We have importin. Cargo binds to importin, okay, because this importin recognizes this nuclear localization signal. Now, this importin and cargo complex will pass through Nucleoporin NPC, Nucleoporin Complex, and here it will interact with these FG nucleoporin motifs. And after interaction with these FG motifs, there will be translocation of this protein cargo complex, and when it is transported to the nucleus, it reaches the nucleus, and in the nucleus we have higher concentration of RAN GEF, which is converting RAN GDP into RAN GTP, and this RAN GTP shows higher affinity for the importin, and when it binds to importin, importin will change its conformation. That will lead to a release of this cargo. Now you can see cargo is released, or our protein is released, and this importin with changed conformation has bound to this RAN GTP, and in this bound state this will exit through this nucleoporin complex by interacting with the same FG motifs, and when it reaches cytoplasm, what was in higher concentration in case of cytoplasm? Yes, it was gap, which will hydrolyze this GTP into GDP. Now this RAN GTP was having higher affinity for this important protein complex, but in case of RAN GDP state, the affinity will be subsequently lower and it will dissociate. Now if it is dissociated, this importin is ready for another cargo protein to attach and transport and continue this cycle. Now similar phenomena will take place in nuclear export. Now to transport proteins out of nucleus, there is something different. The exporting protein cannot just transfer this cargo protein on its own. It needs another protein, which is RAN GTP, to transport this protein out of the nucleus. So this important protein will bind to cargo by a nuclear export sequence. Simultaneously it will bind with RAN GTP on another side. When this trimeric complex is formed, which is cargo protein, this exportin, and this RAN GTP complex, this is exported out by docking with this NPC, Nucleoporin Complex. The interaction with FG motif will take place, and it will be exported outside into the cytoplasm. In cytoplasm there is a gap or RAN gap, which will hydrolyze this GTP into GDP, and in GDP state there will be loose in affinity between these two proteins, and that will lead to release of this cargo protein as well as release of GTP protein. Now this exportin will cycle back into the nucleus, and this GDP RAN GDP will also cycle back into the nucleus. In nucleus we have high concentration of RAN GEF, which will attach GTP in place of GDP. So we have higher concentration of RAN GDP, and this exportin has also recycled back. So these three complexes, or these two complexes, are ready to bind with a new cargo protein through NES and start the cycle again. Now we will talk about nuclear export of mRNA, because not only proteins but mRNAs are also transported from the nucleus. But so for mRNA, the transport is what we say RAN independent. It does not depend on RAN GTPase cycle or RAN GTP gradient. What are the requirements for export? Okay, for exports we need a fully processed mRNA. Fully processed mRNA means splicing has to have happened, and it must be having a 5 prime cap, and it must be having poly a tail as well. And if there is incorrect processing of mRNAs, those mRNAs will be degraded. So for export of mRNA, we need a completely healthy and completely competent mRNA to be transferred. Okay, so the major pathway which is involved in the mRNA transport is NXF NXT1 pathway. NXF is nuclear export factor 1 and NXT is nuclear transport factor 2. So NXF1 is the major factor and NXT1 is assisting the NXF1. There will be several adapter proteins like TREX complex and some other export cargo complexes. There will be FG nucleoporins which will provide selective barrier. Uh, it is same as we studied in the previous slide. We will need some helicases like DBP5 which are located on the cytoplasmic side. They will help in remodeling of mRNPs which are these well processed RNA protein complexes, and it ensures the directionality. There is no specific role of this RAN GTPase cycle for the directionality and energy. So here is the summarized table. We already talked about these major points in previous slides. Now let's come to nuclear export of mRNA. On the left, as usual, we have the mechanism. On the right, we have diagram. First of all, mRNP assembly in nucleus. By this we mean we must have well processed mRNA. It must have 5 prime cap, it must have 3 prime polyadenylation, poly A tail. After transcription and post transcriptional modification, if may, then we have inside the nucleus NXF1 and NXT1. These NXF and NXT will bind on this well processed mRNA, and when they bind, some accessory protein or adapter proteins may also bind, and they will allow the export of this mRNA into the cytoplasm. This export will take place through Nucleoporin Complex and interaction with the FG motifs as usual. On the cytoplasmic side, we have helicase which is DBP5 which is helping in the modeling of this mRNP complex, and it is energy dependent process. ATP is being used. Now the remodeling will take place. The mRNA will be released outside in the cytoplasm, and these uh, main NXF1 and NXT1 will be released as well, and these two transporter proteins will be recycled back and re-imported inside the nucleus through the same NPC nucleoporin channel. Now here directionality is ensured by this DBP5. It also strips off these two factors, these export factors, and ensures the unidirectionality, and it also helps in remodeling of this RNA complex and release into the cytoplasm. So this was all about nuclear export.